Okay, now it's got recorded. Yeah, sorry, I forgot to record that. But anyway, so this is the introduction about several terms here. So let's talk about interface, which is in your notes here. So let me just um, save as notes number 202. Clear this. Let's do interface. Interface is composed of three phases: gap phase 1, gap 1, S phase, and gap 2. Gap 1 is basically cell growth. S phase is DNA replication, and the gap 2 phase is prepare for mitosis. Okay, so right after G2 phase is the mitosis. It has 4 phases. One is called prophase. 2 is called metaphase. 3 is called anaphase. And the 4 is called telophase. Okay? So the mnemonic for this will be TMAT, which is please make a twin because mitosis is making two identical diploid, which is two sets of chromosome cells. Okay? Any questions? Okay, good. So in prophase, we said what got appeared here? In prophase, what got appeared? So let me see. Let me open the other notes here. What appeared in prophase? If you look at this, what turns into what? Yeah, so chromosome appears, right, in prophase 1. So chromosome appears, and spindle fiber also appears. Do you know what is a spindle? What are spindle fibers? Yeah, perfect. Spindle fiber appears, and chromosome appears, and nuclear envelope disappear. Okay. So now, in metaphase, and later on I will just go through this really quick, and I will show you pictures. Okay. In metaphase, chromosomes aligned along central plate. And anaphase aromatase split in chromosomes got split and moving towards 2 poles. Okay. And the last one, telophase is the opposite of prophase. Then can you tell me what will disappear and what will appear? That's right. What will appear? Nuclear envelope. A nuclear envelope will appear. Yeah, that's right. And the spindle fibers will disappear. And by the way, this one, the centrioles, this is only in animals. Centrioles will move to 2 poles. So let me just change colors here. Okay. So let me see. Let me show you. Basically, a picture is better than 1,000 words. So let me just show you the 4 phases. Mitosis, 4 phases. Okay. So this looks like a what kind of phase? Interphase. Because do you have chromosomes? Do you have chromosomes? No, you have chromatin, right? And if that's so basically, can you tell me in this picture what's happening? What got appeared and what got disappeared? Uh-huh. Yeah, that's right. So what else got appeared here? What else is appearing? What else is generating? Yeah, that's called spindle fibers. Spindle fibers, they also appears, right? They also appear. Yeah, that's right. And before we go too far, let me give you a big a quick review for structure of a chromosome. Basically, you have generally two arms of the chromosome. Usually one is long and one is short. Two arms of the chromosome, okay? And the body in the middle is connecting them. It's called centromere, okay? So spindle fiber attaches centromere at a side called kinetochore. So kine means movement. So the spindle fiber will drag the chromosome along, or the chromatid along, through the connection at the kinetochore. Okay. So after a chromosome got duplicated, this is called sister chromatids. Basically, they are temporarily attached together to form a duplicated chromosome. Okay. So these should be exactly the same, ideally. Okay. So at both ends of each chromatid or chromosome, you are having something called a telomere. This determines how long do you live. The longer of the sequences of telomere, the more of the cell cycle that could be duplicated. Okay, so basically chromosomes can be duplicated, but it will become a little bit shorter and shorter because it cannot be duplicated at both ends. And the telomere is trying to protect the end and try to help the chromosome to replicate as long as possible. Okay, so those are called telomeres here. So again, can you tell me what is this structure? And if I got a spindle fiber attached here, what is the attached site called? Kinetochore. Kinetochore. Okay. So each half of the chromosome is called what? Chromatids. Very good. That's right. And they got connected by the centromere. Okay. Yeah, good. Yeah, good. Let's come back to this because this is important. So, and can you tell me if you say the nuclear envelope disappears and the chromosome appears, this is what phase? That's the prophase. And if all the chromosomes got aligned, metaphase, and if the chromatids got separated, and if they reach both ends and form new nuclear envelopes, new nucleus, and the called telophase. That's right. And then after that, you are going to enter what again? That's the action is called cytokinesis. Cytokinesis, and you are going to enter what phase again? You are going to enter what phase again after cytokinesis? Interphase, especially that should be G1 phase. Okay, sometimes you may get branch out from G1 phase to enter G0 phase. G0 phase is a resting phase for the cell, so it's basically the cell goes away from the cell cycle. So a good example of this, so probably you know this, right? if you have a cut on the skin, you may leave a scar there, right? Yeah, that's possible, right? But for babies, it's hard to make a scar because the baby's skin or whatever, the tissue, they can duplicate very well. They are active in cell cycle. So in babies, they may not form scars if the cut is not deep or serious. But for old people, you may form scars easily, right? Understand? So that is because the cells goes away from G1 phase from the cell cycle and enter the G0 phase. So that when you have a cut, they may come back, but they cannot duplicate as well. Okay. Yeah, that's right. So then, can you tell me why do cells divide? Yeah. To yeah, that's right. Basically, why cells divide? First of all, to prevent a single cell get too big and out of control. Okay. Do you know why cells cannot get too big? Too big, less less surface vol surface volume ratio and the nucleus hard to control the cell. So you do not want the cell to get too big. The second of all is to let tissue expand and grow. Okay. And then further on, you are going to let the individual grow and develop. And replace old cells repair damage. Okay? Okay, that's good. Let me see. Let me quiz you a little bit. Let me just quiz you a little. DNA got duplicated in what phase? No, DNA got duplicated in what phase? Is that before the mitosis or after mitosis? In the interphase. It's before the mitosis, right? And it's in what phase of the interphase? So prophase is not in the interphase. Prophase is nothing. Look at this chart. Yeah, that's right. Can you name all the phases in the cell cycle starting from G1 phase? Go ahead. Mm-hmm. 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 Yeah. And the action is called what? The action of the splitting is called what? The action of the splitting of the cell is called what? Yeah, the action itself is called cytokinesis. Yeah. And let me list these down. We are going to finish really quick here. So let me see. You said mitosis. Again, what are the 4 phases in mitosis? Prophase, metaphase, anaphase, telophase. And can you briefly tell me what's happening in each phases? Yes. Yes. Chromatin becomes what? Mm-hmm. Mm-hmm. Yeah. Yeah, that's right. What disappears? So what disappears? In order to have the uh, fibers attached to the chromosome, what must disappear? The nuclear envelope, right? Yeah, good. Go ahead. That's perfect. And what's going on in the metaphase? Chromosome. The chromosomes are aligned in the center of the cell. That's called central plate. Okay. So what's happening in the anaphase? What got splitted? Yeah, that's right. So chromatids got what? Separated, right? Yeah. And what's happening in telophase? Yeah. Uh-huh. Yeah. Okay. That's right. Okay, perfect. That's good. Very good. Do you have any other questions for me? Oh, cool. It is clear. Do you like this? You learned something? Yeah, cool. Very good. You're smart. just review. I'm going to send you these notes right away to your father's email, and I will let him to verify. He's still available, right? He is not asleep, right, yet? Is he asleep? He's still available? Yeah, I will talk with him on the phone. You can just hand the phone to him while I'm sending him the files. Okay, good job today. Nice meeting you today. So if you need any help in the future, just let me know, okay? Yeah. Okay, no problem. Take your time. Hi, Lonnie. Yeah, we just finished. Let me just